Hey, good afternoon, everyone. So welcome to uh, our topic for the single cell RNA sequencing. So you can find today's topic in our Rockefeller University GitHub.io. And in this topic, we will extremely focus on the 10x system. And this course would divided into three sections, including how to interpret the cell ranger QC report and how to do basic analysis by using the browser. And then we'll go through, go through the workflow with data analysis by using Seurat. This very powerful package for single cell sequencing data analysis. And in the third section, we will demonstrate how to process the single cell sequencing data and make a more advanced report QC plot by using the bioconductor packages. So let's start our first section. I assume you have already installed all the packages we need today. Okay, so let's start the first section. Okay, again, we just say then this we are specific, more specific and focus on the tenant system. And so the first thing is to interpret the cell ranger results. What is cell ranger is a widely used software for single cell sequencing data analysis, especially for 10S genomics data. It supports various kind of analysis for 10x single cell sequencing data. It can be used to align to reference genome and make counting. It can be also used to analyze the VDJ changes and their motif. And they can be used to merge different data sets. And we can something is more useful for the genomic center or like our bioinformatics, the bioinformatician. We can convert the BCL file, it's a raw files for the single cell sequencing data into a readable FASTQ files. And for some customized genome version or GTF, we need to use this kind of this function to a function of cell ranger to generate the customized GTF and the genome files for the alignment and counting analysis. So if you go to the genomic center and down your single cell sequencing, you will get a lot of data, including the FASTQ files and some QC reports and the loop browser file. So here I will introduce how to read the QC reports. A QC report you may get for each sample like this. In this sample is estimated 9,000 cells were detected. I will introduce how we determine these numbers later. There are over 9,000 cells detected in this sample, and then the median read per cell is 51,000. This is good, and the median genes detected in each cell is over 1,800. Okay, and for the sequencing quality, you can see here in this chunk, we can see how many reads detected here, and I think most of the barcodes. The barcode means a droplet or a single cell. I think that the validated barcode rate is very high, is 97, and the sequencing saturation is normal, 70%. is normal, and the most reads and the bases are greater than 230. I think that's great for for sequencing data. And if we map to the genome, like this sample is mapped to a mouse genome, uh, let's see, there are over 90% reads mapped to the genome, and there are a few of them mapped to the just a 6% were matched to the intergenic region, and the 20, I think the intronic map to the intronic region, I think is another that there are 23%. I think is fine. Yeah, it's fine to have the percentage a proportion of the region mapped to the intronic region, and most of them are over 60% will match to exonic region and belong to the transcriptome. I think that's good. Okay, so I mentioned how we determine the cells confidently detected in this data set is based on this plot we call the knee plot. So for each class, we rank each droplet or each barcode or each cell. In the I would I would use these terms. All these terms are called means a single cell. So for each single cell, we rank the we rank the we give them a rank by the by the by their by the UMI count that we detected. So the rank one, the first rank barcode, the first ranked droplet is higher than it has a very high UMI count, and it will decrease. The shape is like a knee, so we call it knee plot. Okay, and which one we have to pay more attention is that when the rank when the rank when we go through the right side, the UMI count will be decreased. When it comes decrease very quickly, that means the read count would drop very quickly. So that means the critical point to determine a cell is valid or not. So after like in the right side of this plot, this barcode only contains few read counts, so that means that would be just a just an error or random error or just useless cells. So we will remove it. So that's why we can determine the cell barcodes. That, that's why we can determine the validated cell barcode is around 9,000. We will introduce more details in next sections. Yeah, and determine and show how exactly to calculate it using some droplet utility, this package. So we can estimate, based on this knee plot, we can estimate the number of cells using over 9,000, and the fraction breathing cell is 76%, so good. Mean calculated mean reads and the median gene per cells, etc. Okay, so you can see here in this overall data set, in the total cell, in the entire data set, we can detect 19,000 genes, and for each cell is around 1,000 detected. Okay. So the last chunk is about some sample information, like the sample name, short description. which genome we used, and which library we use. Like this one is a 3' and priming library, and which genome, which cell ranger version we use. Right now, the latest cell ranger version is 6. Yeah. Okay, so it's a brief description about the the QC report, the QC summary about the QC summary generated by cell ranger. Okay, so it can also in this report is also included the clustering, clustering cells and the plot tSNE and the meta tSNE plot like this, and then they also show several the potential marker genes for each cluster. Okay, so that's the basic QC report for the cell ranger, and we also and this after the analysis, the cell ranger can also produce a loop file. It can be processed by loop browser. So let me check. Switch to the loop browser. Okay, if you have experience about the loop browser, and it looks like this. When you open it, when you open the loop file in this browser, you will see a tSNE plot displayed in the middle, and there are several information here, like the default setting is a tSNE plot, and the cluster cells by the graph-based clustering. Okay, and here they also shows the potential marker genes for each cluster. You can see for the cluster 1 compared to other clusters, there are this genes is one twofold change, and then their p-value is very low. For the other, for the class 2, is this genes seems the good marker genes. Okay. So the yeah, for the loop browser, we can do some changes like our customized analysis. We can produce, we can regenerate the loop browser with the tSNE plot or the UMap or customize the UMap. So you can see the usually I prefer use UMap because usually it makes more differences between clusters. And we also provide you labeling like we can use Seurat clustering to make we can label cells with our customized Seurat clustering. Yeah. So we can make a customized a loop file, and if we want to test some marker genes we already know, like this gene, like this data set, it maybe several cell has a MRC1 expression, so you can switch to the gene feature expression tab and just type your marker gene here. You can see the cell expressing MRC1 is accumulated in this cluster. Okay. And if we want to test another genes like a PTPC, yeah, it's another kind of marker genes. You can see the cluster here is shipped here, and if we want to test like some keratin to show up the epithelial, Also, you can see this cluster might be the epithelium. So that's a yeah, that's an it's a very the loop browser provides you a very easy to operate it and very straightforward and the intuition uh, and the very intuition to analyze your data. So yeah, that's a good start to understanding your single cells. And we can also based on these marker genes, we can also make some subclusters. Yeah, like we can use add the filter that we see in the not, not the threshold is larger than one. So it's a look to features. Okay. I try to make some clusters. Yeah, maybe I can show you. I'm not really use this a lot. So yeah, maybe I can skip it. Okay, sorry. So make a summary. There's the yeah, make a summary. There's a loop browser that is very useful if you want to have a quick look about your data. Yeah. And if you want to make, if you have interest in how to process in this loop browser, you can go to you can go to 10x Genomics website. They have several tutorial to help you to figure out. But here I will put more attention to use a further analysis, with Seurat and the bioconductor packages. So we will describe more details in next two sections. Okay, so back to our slide. So here we want to show the customized analysis that we can provide to our customers. Okay, so I will use this article to as a demonstration. Is published on eLive last year, and okay, so um, we we analyze the single cell sequencing data for this paper, and this is what we can show. This is what we can provide to you. We can make the we can make a customized loop browser and tSNE plots like this. There are two different cells, different data sets aggregated. So we can separate these data sets and calculate and classify all the cells, classify all the cells, and make a marker gene, calculate the marker gene for each cell type, make an heatmap, make a heatmap like this, and then we can also provide a shorter time analysis to yeah to demonstrate the different lineage differentiation trajectories for each sample. Yeah. And then based on this information, we can compare gene expression, differential gene expression. We can compare differential gene expression between two data sets, and for each cell type between two data sets, and calculate the single cell gene set enrichment analysis for each data set, and then provide a feature plot like this. So this is our regular analysis for the single cell sequencing we can provide. Yeah, we are also uh, we are also we are also trying to do some like the RNA velocity and other customized analysis, all kinds of the customized analysis. So that's what we can provide for you. Okay. So for the next two sections, I will go through our regular analysis workflow for single cell sequencing data analysis. You can download the data because the data is too big to upload on GitHub. So you have to download from either the uh, either GEO or the GEO database or box. So like the box link, you can see the control sample. I will use a control sample to do all the course, and for the knockout samples and for the exercises. So it contains the two different folders. It contains two different folders. Is for the single is for bioconductor one or the Seurat. It's because their naming is a little bit different. Basically, they are the same counting metric. So is there? So in this session, we briefly introduce how to read the cell ranges basic report and show the basic operation for the loop browser and show the customized regular analysis for us that we can provide. So we are started the next session is about the how to analyze the data with Seurat. Okay, so in this session, we will focus on the Seurat to analyze data with Seurat. It's a very powerful package to analyze for single cell sequencing data analysis. They can also support many common analysis for single cell RNA-seq, and it can cause connection with many useful packages like in the monocode to do the pseudo time analysis and something for some others for RNA velocity, velocycle for RNA velocity, and for function of they can also convert the object to support the function of differential gene expression analysis packages. All of the conversion data conversion can be done by Seurat wrapper. Okay, in this session, we will demonstrate our regular analysis workflow for single cells RNA-seq data analysis. So it includes how to read the cell range results into a Seurat object, how to generate the QC report, QC plot, evaluate the cell cycles, and normalize data and do clustering, how to find the marker genes for each cluster, and some advanced visualization. Okay, so if you download the data from box, you can put on your directory, and then we can use this command Seurat from Seurat read 10x to read in all the folder. The folder should come sorry. I mean, in the folder we must contain three different files. One is a barcode, the, the barcode information. Features means the gene ID information and a matrix. So all of the three files should be available should be contained in the folder. Okay, so you can use the read 10x this function to read your matrix in read your matrix and then use create Seurat object to load the content matrix into the Seurat object. And here we set the minimum cell at least has 5 reads, and their features is larger than 200. And then make some we will make a additional labeling like this data set is the code control. If you have a multiple data sets, you mustn't assign your data set well when you load when you create the cell range objects. And this is for renaming the barcode. Cell barcode is not necessary; it's optional. Okay, so the next one is about uh, in the in additional step we will calculate the mitochondrial contents in each cell. You can use this function to this command to calculate the mitochondrial contents for each cell. That means calculate the percentage of certain features. The feature must the pattern of the features uh, contains MT dash. So you can find all the features contain this string and calculate the proportion in all the in the entire cell in the entire count. So it's a usually the single cell sequencing data is very large. It's not easily to uh, for demonstrate in the course. So in this course, we just take a small subset of the for for demonstration. Okay. So we can see what the contents in the Seurat object. We can use the metadata to show the content, to show that each to show the general information for each cell. Okay, like this one, the row name is the cell barcode. Is each cell barcode, and then for this cell barcode or for this cell, it contains over 7,000 reads, and there are 2,000 genes were detected. It belong to the control group, and there are 5% over 5% of the reads belong to the mitochondria. Okay. Okay, why do we need to know the mitochondrial content? I will explain later. It's a very important question, especially for the single cell RNA sequencing. It can be also useful for single nuclei sequencing, but we don't mention single nuclei at this moment. So you can get in the Seurat object also contains the content matrix like this. Then for each cell, each column is for means each cell, and each rows means the genes. So like for this cell, they got one count for this genes and one count for this genes, yeah, etc. And we also have the after normalization or scaled, we can also get a scaled matrix like this. Well, I will describe how to scale or normalize data because that's another important step in the when we normal when we do the normalization. Okay, I will discuss more detail later. Okay, so there are several important criteria to evaluate the data. So we have to consider this and this uh like these three data that like these three features like the recount means recount per cell, that means yeah, recount per cell, or gene count per cell, or the mitochondrial content per cell. And you in Seurat you can use VLM plots. That means a volume plot this function to to evaluate their distribution of these three different features. Okay, so you can see here the, some of the genes identified. The major proportion of genes identified is here, like more than 2,000, and the count for each cell is around the median of the count per cell is around 10,000, and we can see several cells contains extremely high percentage of mitochondrial genes, and most of them are low. So for the given three different features, we can make some comparison scatter plots like this. So in this plot, we try to compare the read count and the gene count per cell, and you will see they are highly concordant. Yeah. But in this plot, we have to pay more attention about this region. This region has that means in this cells in this region means they have a extremely higher gene count and higher read count. So it's probably I mean probably they are doubled. That means several cells, two or more cells clumped in a single droplet. So so in this droplet they has more gene detected, more reads detected than other distributions. So I will introduce how to evaluate the doublets and remove the doublets uh, in the section 3. Okay, and then in this scatter plot, we compare the recounts and the mitochondrial contents, and you will see this pattern. It means uh, many of several of the cells has very low recounts, but they has a very extremely high content of mitochondrial genes. What does it mean? Uh, it usually means they are cell debris. So they has a lower nuclear nuclear genes, but they have a very high count of mitochondrial genes. Yeah. So the cell debris is not we expected to get. So we would remove this kind of cells. Yeah. So that's why we need to evaluate the mitochondrial content for each cell. It's just to remove the cell debris. It's a quite important issue because after cell ranger version 2, they make a trade-off to make more alignment and get more genes to count in more genes, and so they wouldn't forcibly include the cell debris inside. So they would use this kind of strategy to remove the cell debris. Okay. So here I just introduced in this plot the cell debris has there's uh, characteristic of cell debris is a very high mitochondrial content and a very and relatively low rate count. So we often set a cutoff to eliminate the cells with a very high mitochondrial content. But I usually use 10%. But if your research target is an activated immune cell or the muscle cell or even the muscle cell that was extremely much higher the mitochondrial content, we would consider this cut will reset this cutoff just to fit your research in your research aims and objects. So that's before filtering, we got the routing part like this, and after filtering, we remove the cells with the cells with the mitochondrial content is larger than 10%. Okay, it's a scatter plot before and after removal. Okay, so it's a general step to load the cell ranger data counting metrics into a Seurat object and make a simple QC to evaluate your data quality and to remove just simply to remove the cell debris. Okay. So next step we can and then we want to introduce how to determine the cell cycle phases. Okay, in Seurat there has a very simple way to a very simple way to determine cell cycle phases. They use a marker gene. They have a bunch of marker genes for S phase and G2N phase. Is stored in the cc.gene list. If you load the Seurat library, it will be automatically included. And this gene list is based on the human data. Is based on human data. So in this case, we use our research target is mouse data. So we have to convert the mouse human gene symbol to mouse gene symbol by using the boundmart. Okay, so here is a simple function to convert the human gene symbol to the mouse gene symbol. And if the and then this line is about we converted the S genes, the marker genes for S phase, and this is a conversion of the genes for G2N phase. Then we can just score in our match our cells and calculate estimate their cell cycle phases by using this simple function called cell cycle scoring you have to provide your Seurat object, and the genes we use the gene signatures for S phase, the signature for the G2N phase. Okay, so you will get they will add a column in your object called they will add several columns. I mean they will add a column to for describe your S phase score. The G2N score is based on the signatures is estimated by the signatures, and then they would judge the cell cycle phases by using this signal by using this score. So the first like this cell its score is this S base score is minus, but is G2N is plus. So in the overall distribution, after comparing overall distribution, they would consider this cell is in the G2N phase, and the similar reason this in the similar reason this cell is also a G2M in G2N phase. Okay, so we can simply calculate the cell cycle phases in our data set, and you can see here there are 250 cells in the G1 phase, 200 in G2M phase, and 227 in A phase. You wouldn't be considered if this proportion is good or is something wacky, but this that's because this skin cell is under is harvested in the mouse. I remember it's from a mouse is developing, and so it would expect that there are more cells in the G2M and the H base. So I think it is fine. Okay. So we can, based on this table, we can make a bar chart like this. Okay, that's how we evaluate the cell cycle phase. So the next part is to regression and or normalization, and then clustering all the cells. So the first step is to normalize our data. Which one we have to rescale or normalize to is the percent of the mitochondrial content, because this is not our interest. So we would ignore we want to regression to the percentage of mitochondrial content and the S score and the G2M score, and plus the phases, cell cycle phases. We don't want this as we don't want to evaluate the genes responding to these changes. Okay, so we can use this function called the scale data. So select your Seurat object and choose the variance. The variance you want to regress or want to normalize to ignore it. Yeah, you can set it here. So it can generate another scaled data matrix in your Seurat object. Then after scaling the data, we have to if we want to make the next step is to make a clustering. So we have to calculate the principal component. We have to run principal component analysis first. So we use a run PCA to process principal component analysis. We just counting the first 13 principal components, and then how many PCs we have to can how many PCs to represent your data is to represent your data. So we use elbow plot to find out how many pieces we have to take. So use the elbow plot, this function, and you can display the pieces. And if you can see here, this plot is a little bit like an elbow, and if you see that they will drop them very rapidly if they becomes they turn if the curve turns flat, that means that means the pieces is the best pieces we have to take. So in this, so in it's it's a little bit arbitrary. So in this case, I would take the 15 pieces to do the following clustering analysis, for clustering and the following analysis. So here we set we will take the first 15 principal components and finding their neighbors, do the finding neighbors and do the and compute to the nearest neighboring plot, and based on this plot, we can find out, we can distinguish how many clusters inside in our data set. If you want to get more data sets, you can fine tune this. If you take a larger resolution, you would expect to have more clusters identified. If you don't want to separate your data into too many clusters, uh, you can turn down this. You can use more smaller resolution numbers. Okay. So for this case, we use the default setting, and we use a default setting to identify clusters. Then we can run the UMap to generate a UMap plot and generate a tSNE plot. Okay, so after that, we can use Dimplot to display our UMap plot. In the function, the usage is like this. We use a Dimplot, select your select object, and select the reduction type to UMap. If you want to do tSNE, you can select the tSNE. If you don't assign any reduction, it will be taken UMap first. So in this card, so in this data set, it can be separated into 6 clusters, and their UMap distribution is like here, like this. If we turn the tSNE, it's like this. Sometimes you can I would suggest that plot tSNE plot, do tSNE plot and both tSNE plot and the UMap, and see which one is more fit to your explanation, your your hypothesis, or your explanation is good for explanation. Both of them are the based on machine learning, different type of algorithm of machine learning, but I think both of them, these plots are good. Just depending on which one is more fit to your research. So here we show how to make normalization, data normalization, and clustering, and make a general plot to display the clustering, to cell clustering. So the next step is to identify marker genes for each cluster. So the first thing is to set our identities for your Seurat object. We need to make sure it's based on the identity is based on the Seurat clusters, so they would calculate they will when we run the next like the finding all marker this command, they will search the marker genes by clusters. If we set by other factor like we set by the data set, you will find all the marker genes in different data sets. So it's a very simple and useful command called find all markers to identify all the marker genes for each cluster. Usually we will just display the positive one, and their minimized percentage means the minimal the proportion of cells expressed in this given genes is the minimal proportion is 25%, and we will also get set the log4 changes threshold. I remember is 0.1. Okay. So based on this setting, they will calculate the markers for each cluster, and they will turn back the data frame like this. So it means for this gene is a marker gene for class 0, and is expression in how many cell expression in class 0 is I mean there are 81% of the cells express this gene in class 0, and 40% of cells in other clusters express these genes. And we got a p-value and the log4 changes. Okay. So once you get a marker genes for each cluster, you can compare to other research or something else to differentiate or just annotate the cell types for each cluster. Okay. For for example, we can if we so you can get this class marker. This object contains all the markers, all the cluster specific markers. For a plot for visualization, we can just take the top 5 marker genes for each cluster by using this command, and then you can get this kind of tables. Yeah. It's for the following analysis. I will show you late in that section. Okay. So for the next part is for the advanced to do some advanced plots with Seurat. Okay. So this is a Dimplot UMAP. This is a UMAP. We can display the UMAP with a Dimplot, and usually I will set the point size as 0.2. That means it's because in the default setting the point size is a little bit larger. So if we put it in we put it point set a point size with a smaller point, we can see more clearly, especially when we add the labels of each clusters like this. We can directly label our clusters on the point on the point, and then we can remove the legends with this adding this command no legends. So it's much more straightforward. You don't need to compare the legend and the colors. And as we mentioned, we just took the top 5 genes for each cluster and then display the heat map like this. So for each markers, they would the um, the gene expression, the scaled gene expression for each cell is an is colored from the purple to the yellow. So it means for these genes, this genes is expressed mainly in class 1 and in several class 2, in class 0 and class 1. And like these genes for class 2, they were very highly specific. Yes, like the SOX9 and the KRT17, they are very extremely specific in expressing in cluster 2, but not in other clusters. They are also We can also find the very good markers for cluster 3. They are only expressed in cluster 3, but not others. It seems the cluster 3 is epithelial cell, and the cluster 2 is, I remember that the SHH, some, that's a SHH activating cells. Yeah. Oh, no, sorry. It's a SOX9 activating cells, and this one is another lineage. Is Sonic Hedgehog expression cells in the cluster 4. Okay, so we can also display the top markers for each cluster in the feature plots like this. So you can see the keratin one is expressed in this small proportion, and the PTH-LH genes is only expressed in this proportion, and Sony Hedgehog is only expressed here. And then for keratin 14 is expressed in most of the cells, but is mainly is expression more in the central, in the central, and then several genes, several cells express the sample genes. And this is another type. We can display the marker genes for each clusters, and most of them, if they are not expressed, they would skewed in the left, and if they are expressed, like this one, so this means the KRT1 genes is expressing is much expressed in cluster 3 and cluster 5. So for this gene is expressed more in cluster 2, and for this gene is expressing in most of the cells, but is expressed more in this kind, in the 0 and 1, 0, 1 and 5 cluster 0, 1 and 5. And for this gene is expressed more in the class 1, and the class a little bit more enriched in class 5. Okay. So that's when we get the marker genes, we can do some more fancy cluster like fancy protein like the feature plots and the rich plots. Okay, so we have already know the cell cycles. We have already estimated the cell cycle phases for each cell, and we know that we already clustered our cells. Is there any enrichment of the cell cycles, the cells in different cell cycles? Uh, compare how we compare the cell cycle phases for each clusters. So we can just extract the information from the meta tables, make a table to calculate the Seurat clusters and the cell cycle phases. And then you can generate a plot like this. So you can see in most clusters, and they have the similar proportion of uh, cells, similar proportion of cell cycle phases. But in cluster one, it contains much more cells in S and G2M, but only less of samples has the G1 phase. So that means this cluster would be the very highly extremely in the extreme cell cycle activation. So the next, so we this would remove these clusters, yeah, to eliminate the cell cycle events. And then, so finally, we can save the Seurat object into a RDS file. Okay. So the the next slide is just to introduce the Seurat this packaging is uh, it can be converted. The Seurat object can be converted into different objects for further analysis, like monocode for the shorter time analysis. We have done this, and Veracyto for SC value for the RNA velocity. We are still trying this, and if we want to get more information about Seurat wrapper, you can go to their GitHub site and get more information. And you can see how many they are supporting many packages. So it's time to uh, exercise. So in this exercise, you can download the data from the box, or just take our examples from the GitHub site, and then the first thing is to load your object into load the content matrix into the Seurat object and calculate the mitochondrial content for each cell. Then make a basic QC plot like this, and a scatter plot to evaluate the potential doublet. That means that the cells containing extremely higher gene count and post gene count and the transcript count like this. Okay, make a make a plot like this. The transcript count and the percentage of mitochondrial content, and then and then evaluate the potential cell debris. I mean, like this extremely high content of mitochondria genes, but very low transcript counts. And then remove the cells with the percentage of mitochondrial content larger than 10. Then make a cell cycle analysis with Seurat default function. After that, make a normalization and the cell clustering. Try to regress to the mitochondrial content and the cell cycle. And then make a PCA analysis. Select how many PCs we have to select for further analysis. Then make a clustering and make a UMAP plot. The next part is to evaluate the marker genes for each clusters and make a heatmap. Then select the top marker genes for each cluster and display in the feature plot and the ridge plot. Okay, so we will take, right now is 3 o'clock. We will spend 30 minutes to finish this exercise, and we will come back. I will say we will come back at 3:45, and we will have a discussion about this exercise and then enter the next section. Okay, let's start our exercise. And for and you have to the first step is optional. You can start from our data, the whole data set from box, or download from box, or using our DS file. I have already make a subset. So if you from a box, just from the box, you can follow in this section to assign use read 10x this command to read the whole to read the 10x result into a counting matrix and make the Seurat object from the counting based on the counting matrix throughout the cells with too few counts and too few genes detected, and then use this command to calculate the percentage of mitochondrial contents per cell. Then we can make the basic quality control plot. So we need to use the volume plot to plot in these three vectors, these three features, like in the gene transcript count per cell, gene count per cell, and the percentage of mitochondrial content in each cell. Okay. And then the third question is a method scatter plot. A scatter plot, the x-axis is the transcript count, and the y-axis is the gene count. So we got the plot like this, and then which one is potential doublets? We can see we can estimate the cells with extremely high genes, extremely high transcript count and extremely high gene count would be doublets. And we can make another scatter plot. The x-axis is the transcript count, and the y-axis is the mitochondrial content. So which would be the potential cell debris It means the depth. In the depth, they have a they have a higher they have higher percentage percent of mitochondrial content, but with low transcript count. Then we will select the cells which we will remove the cells with the percent of mitochondrial content larger than 10%. Okay, so the next one is for the cell cycle. Again, we can use the the gene features provided in the Seurat um, and the first thing we have to convert the gene features into from human to mouse, and then use this command cell cycle scoring to score each cell to calculate the S-base score and the G2M base score, and then based on this score, they can determine the cell which cell cycle phases is for a given cell. So this information would be stored in the metadata, so we can make a simple calculation about here. Okay. And the third part is for normalization and the clustering. Before normalization, we have to do this. Sorry, we have to do this. This step is to find out the variable features. That means the a bunch of genes which can be the most difference, making most difference in this data set. So we have to find out the most variable features, and then based on these features, they can scale data. They scale the data, and which one we want to regressing or normalize. We have to in this case we have to regress to the percentage of mitochondrial content and the cell cycle event like the N score, G2M score, and the phase. Okay, so based on this normalization, and we will run the principal component analysis like this, and use elbow plot to estimate how many PCs we have to use for following analysis. I think in this case, I would choose 10 the first 10 principal components. So we found the neighboring, use found neighbors and found the clusters to identify to clustering the cells, and then use run UMAP to generate the UMAP plot, to estimate the UMAP plot and the tSNE plot. Then we can display the UMAP plot by using Dimplots, and we can set the reduction equal to the UMAP. So we get a UMAP plot. There are 6 cell clusters identified in this data set. Okay, next part we will try to select the marker genes for each cluster. So the first to do this, we have to set our active identity into the Seurat clusters, and then use find all markers to identify marker genes for each cluster, and you will get this kind of table. Then we can take the top 5 genes top 5 marker genes for each cluster and then make a heat map like this. Okay. Okay. While we take the top 5 genes of each cluster, we can take the top 1 marker gene for each cluster and make a feature plot or a ridge plot. Quite simple, just take a feature plot. You can see these genes is only expressed a small proportion here, and this gene is expressed here. Yeah, electron, and we can also display in a ridge plot. Okay, that's this exercise. Yeah. So the next we will go through. We will discuss. We will go to the section 3. It's about how to manipulate the uh, single cell data with bioconductor packages. Okay, so there are more and more bioconductor packages supporting single cell data analysis, and they also provide integrated workflows, and you can go through the if you are interested, you can go through the entire workflow and the course in this website. Yeah, it contains a lot of packages, how to analyze the data and give you a lot of examples. So that's very useful one. Here we just describe some advanced analysis and something is different from the something Seurat is not easy to do. So in this session, we will discuss how to differentiate empty droplets, how to estimate the ambient RNA contamination and remove them, and how to identify the droplets, um, then check any compound, check is there any compound effect. Okay, so in the beginning is still the same, how to load the data into the single cell experiment object and calculate the empty droplets. So here, in the first step, we will use the function, we will use the package, use a library called the droplet utilities. So if you download all the data from the box, and you can direct your data, you can direct your data folder, provide the file directory to the cell ranger file folder, and then use this function, read the 10x counts to read all the data into the single cell experiment object. Okay. In this case, we just take some small subset for demonstration. So if you read the if you read the data 10x data into the single cells experiment object, you will get an object like this. It contains how many, this is genes and this is cell barcode gene detected and a cell barcode. We have a metric counting metrics and the gene names and including gene ensemble gene ID and the gene symbols. Then for the column means the cell, we have a cell barcode and their sample names. Okay, like this. the cell barcode and the sample name. And for gene information is in symbol gene ID and the gene symbol. Okay, so the first thing we have to do is to access the UMI count, the read count, the transcript count in each droplet. So we have to use this command barcode ranks to, to calculate the UMI count in the single cell experiment object, and you will get this kind of data frame. Is for each cell is ranked by the UMI counts. So it's like the knee plot. The x-axis is the cell barcode ranked by their UMI count. The y-axis is the UMI count in each droplet, and we will find out that 2 points is very important. Yes, the first is an inflection point. That means when the plot is decreasing rapidly, started decreasing rapidly, and the knee plot is a cutoff, means the cutoff UMI count to differentiate cell value for analysis. So the knee point uh, de determine which cell is available, which is a cell and which is just an empty droplet. So we can plot like we can make a plot by using following script and plot the rank the rank and barcode and the count, then then point out the inflection point and the knee point, make some bigger regions, so you will get the figure like this. So for the blue dashed line means the inflation point. So you can see that that's the that means the start decreasing rapidly, and for the knee plot it means the turning point of this plot. So that means they are above them above the cell with the UMI count larger than this knee plot would be considered as validate, and for these cells they would be considered as an empty droplet. Okay. So it's very similar to the QC report to the knee plot in the cell ranger QC report. Okay, so we can identify the empty, non-empty droplets by using two methods. One is to just assign the lowest count per cell, like in the Seurat. In the previous section, we assigned the 200 as a lowest count per cell, and for this in this case we just set the 100, and then we can use the and for the this count and for the sorry the droplets the utilities they can use this on empty drop function to to estimate the probability of a given cell is an empty droplet or not, and then they will give you a FDR, a p-value, and the just a p-value. So if you go through this function and okay, the test the ambient is used for the would be generate another information for ambient RNA contamination. We will discuss later. Okay. So if you run this function, you can you can get the data frame like this. For each cell, then p-value, the p-value means it's empty. It's an empty droplet or not. If they are significant, the FDR is lower than 0.001. That means it's a real drop, it's a real cell, not an empty droplet. And the limited means it's past the lowest count or not. So we can see here that if we compare the two methods, if we compare just the low set up the lower count or set up the by the or estimated by the uh, probability, we can get this kind of a table. Most of them are agreement, uh, agree to each other, and we only take this proportion of cells as a non-empty droplet and for following analysis. Okay, here we just want to display the check the distribution of the non-empty of the FDR for the the p-value distribution for the non-empty non-empty it's a non-empty droplet. Sorry. Then we have to make a subset of the non-empty droplets. We just choose the droplets with an FDR less than 0.001. Okay, so next step is to normalize and clustering, just like is a very similar as a Seurat, similar strategy as a Seurat. So we can follow in these chunks, we can do the quick normalization, counter normalization, make a quick clustering, compare summary factors, and then make the convert the normalized count to log scale. Okay. So we will get a new assay called log count. Then we have to identify the variable features, just like we said in the exercise. We need to find the variable features. So we have to model in the genes variables and then get the top gene features. Okay. So these genes would be the variable features we for the following analysis. Then we can make a tSNE plot and a UMAP plot by using these chunks. We can evaluate how many PCs we use, and then using the PCA plot, use the PCA to calculate the PCA result to calculate the tSNE plot and the UMAP plot. Okay, so they are a little bit different for the clustering between the bioconductor one and the Seurat. In bioconductor is based on the graphic-based uh, clustering. So we have to build out the graphic clustering, and then yeah, we have to run this clustered command to make a cluster and convert these clusters, uh, label them clusters by using this command. So we will get if we go to see the finishing this chunk and then we go through the code data. That means for each cell, information for each cell you can find. For each cell, we can get one more column called the label that's equal to the clusters we mentioned in the Seurat object. Okay. So if you plot the UMap, you can make a UMap plot by using plot UMap and colored by the labels. Okay. Here we have 7 clusters and with different labeled with different color. We can also make a tSNE plot. Okay. So for the ambient RNA, okay, so maybe we have to stop here for a moment, and I would, yeah. So in this part, we know how to load the file and how to remove empty droplets and make normalization and clustering. Okay, before the ambient RNA, maybe you have just a 2 minutes break. I'll be back by 2. Sorry, I'm back. So, uh, so after the basic analysis, right now we have to we will enter in another section called how to demonstrate how to remove ambient RNA. So the ambient RNA means the cell-free RNA contaminated in the droplet. It might be because if one cell is ruptured before when we separating the cells, and their RNA is falsely included in the droplet, so we will detect this RNA. For example, if a red blood cell is ruptured, the hemoglobin RNA would be disseminated in the whole solution. If this RNA is forcibly forcibly enter a droplet, we would consider this cell is raising the hemoglobin, but it's not true. So we have to remove this kind of RNAs. So as we mentioned, that we do one step to estimate the ambient RNA in the empty drop, and in the empty drop that function. That's because the most ambient RNAs would contain the most easily to find out the ambient RNA is in the empty droplet. One most of the ambient RNA would be easily identified in the empty droplet. If that occurs in the non-empty droplet, we can't differentiate. So we need to extract the information from the empty droplets. So we can go through this one. This object is generated by the empty drop estimation, and if we look at the potential ambient RNA, we can get the genes, a bunch of genes like this, and then a probability of ambient RNAs. Then we can use this function from the scattered this library called remove ambience, and then we can assign our counts, our counting metrics, and the ambient RNA we want to evaluate, and the groups inside the groups or the labels inside in this object. Then we can we can estimate each gene is a given gene is an ambient RNA contamination or not. Then we can, after that, we can make a log change normalization. So take an example, the hemoglobin A1. Hemoglobin is a most common ambient RNA contamination. So we can use this as a target. We can evaluate before and after we remove the ambient RNAs. What would happen? So we have to because for the single cell experiment object is mainly use the ensemble ID. So we have to convert our gene symbol into ensemble ID. And we can use plot express this command to evaluate the before we removal and after removal, and just focus on the gene features. Just focus on the hemoglobin A1, and we will get this plot. You can see here before we remove, they are all clusters containing several cells expressing HbA1, but it's not possible. So it wouldn't not possible. So after removal, we can see only a few. We can see most of the clusters with without an HBA1, without detecting HBA1, but only a few of the cells in these two clusters are can detect these genes. So that means that probably, I mean, probably we don't take, we don't calculate, we don't evaluate very seriously. I think it's probably like it's a doublet cell, a normal cell contain clump with the RBC, so they clump together, so we can find this. After removal, we can see most of the cells, most of HbA1 contamination would be removed. How about another genes? I took the KRT17 is not the it would not be the contamination because the entire skin cell would express a certain amount of KRT17 genes. So we analyze before and after MBNN removal, we don't see any changes between before and after removal. So that means after this analysis, we can only remove the ambient RNA, but not affect the normal RNAs. That's a good sign. Then, since we remove something, remove and recalculate the counting metrics, so we have to rerun the normalization and the clustering again. Okay. And maybe we can save the RDS file. So the next step is to evaluate the doublet. In the section 2, we estimated the doublet is the cells with extremely high read count and the gene count. And for the bioconductor packages, they have like a scRNA, they have they can estimate, they can make the estimation and to evaluate this cell is this droplet is a doublet or not. So it's just a simple command called compute doublet density. Doublet density is the evaluate the doublet, evaluate the count, the count distribution, and is this outlier or not. Then convert it into based on this distribution, they can calculate the doublet score, and then we can input this doublet score into our object. Okay, so for quick evaluation, we can put the doublet score in the UMAP. You can see, I think as you see here, most of the cluster containing both the high or low doublet score. That means no doublet is enriched in any cluster. That's a good sign. If a cluster is enriched, if the doublet is enriched in a single cluster, that means that the cluster is based on the doublet. So we have not the cluster would be removed because most of them are doublets. So we can also evaluate in the violin plot. So we can see the same trend, no single cluster hovering expressing higher doublet rate than our higher doublet score than other clusters. Okay, so we can just remove the 95, set the cutoff, of the doublet score, and then remove the cells over 95% quantile of a doublet score. Okay, just calculate the cutoff value and annotate the doublet, and then remove the doublet, keep the remove the doublet. Okay, so that's the step for to evaluate the doublet and how to remove the doublet. Okay, so make some QC plot after clean runs. We can also calculate the mitochondrial contents and the gene detection and the reads per cell by using this command. First thing, we have to identify the mitochondrial genes, the gene symbol of mitochondrial genes, and calculate it and calculate the mitochondrial content and get these three information by using the add per cell QC. Okay, after that, you will find your data set has 3 more columns containing to describe the genes detected and the recounts per cell and the mitochondrial contents. So based on this, we can make a volume plot for each value. So for post clean runs, we can use a product called data to make the volume plot like for each cluster like this. So this is our object. It's our single cell experiment object. The x-axis is the clusters, and the y is the sum. Sum means the total reads per cell. Okay, and then we make the colored by the clusters. We can also make the plot of the gene count by using this command, and evaluate the mitochondrial percentage by using this command. Okay. So we can see most of the cells were contain their mitochondrial content is lower than 10. So I suggest not to do more more eliminate more subset to eliminate the mitochondrial contents. And we can also plot the scatter plot. We can use the code data to make the scatter plots like this. And you can see that these parts are similar to the Seurat one. Okay. So the next is to evaluate the variance explanation. We have several variables like the clustering and the mitochondrial content, and the doublet score, or the recount, or the gene detected. Which one would explain more the most of the variance is very important. As we would assume our variance is majority from the clustering. Okay, so in the scatter library, it provides a function called the get variance explained to evaluate which one would be the most important. So if you type this function and set up the variable we want to test, we will get this kind of figure. Yeah, you can see most of the variance can be described by the label, that means clusters, and for others is just can describe less than 0.1% variance. So it means this analysis is reliable. If there are any confounder effects, you will see the both of them, or even three of the factors would be have the same value, would be explain the same percentage of variance. If a confounder effect is identified, is a vector is identified as a confounder, we have to do more analysis to remove the confounder effects. But it's not we would not describe it in this section. Okay, so it's time to exercise. So for this exercise, you can download the data from Box and load the library, load the data into the. You can load the data into a SCE object, and then the first question is to draw a knee plot and identify the inflection point and the knee point. Then identify the non-empty droplets by using the limit as 100 or the FDR cutoff less than 0.001, and then make the normalization and the clustering and plot the UMAP like this. The fourth question is to evaluate the ambient RNA contamination and try to evaluate the by using hemoglobin A1 genes, and recluster after removing ambient RNAs. Then the sixth question is to removing doublets, to calculate the doublet score and remove the doublet. To evaluate any enrichment of doublet score in the clusters. So the next part is to estimate mitochondrial content and the read count and the gene count for each cell, for each cluster, for each cell, and the plot, rather the violin plots, like these three, and the scatter plots. Then evaluating which one to find out the factor to contribute to the majority of the variance, like make this part. Okay, so that's for section 3. It's 4:30 now, so we would discuss this section around 5, and after that I plan to describe more details more detailed information about the loop browser, I think I gave several important or interesting functions. So if you are interested, you can join us in the at after 5. Okay. Okay. If you have any question, please leave a message in chat. Okay. We can go through the, and let's go through the exercise quickly. And again, you can choose the data set from box or just take our subset from in the data folder. And the first question to make a new plot like this. So can make a new plot. So the first thing is to rank your barcodes by count, and then remove the duplicate, remove the uh, duplicate, duplicate symbols. Then you can make a plot by this function, and add the line, and then add the line to point out the inflection point and the knee point. Okay, but you can use empty drop this function to calculate to estimate the non-empty droplet, and we will choose the FDR less than 0.01. So uh, after the estimation, you will get this data frame, and you will get the FDR, and then it's a partial limitation or not. So finally, you will get this kind of a table, and you will find out that there are 2,100 cells partial filter, and they are on non-empty droplets. So the next step is to do normalization and clustering. Okay, so we can make the normalized counts by this function, and then modeling to identify the most variant, the most variable features, and by these two functions to modeling the gene variants, and choose the get the top gene features. Then evaluate how many PCs to describe this data set, and then process UMAP analysis. Okay. Then cluster cells with a graphic based method. So finally, you can generate the UMAP plot and label with the cluster color, label the colors according to their clusters. Okay, next part to evaluate the ambient RNA. So we may need the object generated in the previous section to evaluate the empty droplets. It will calculate the ambient RNA and their probability. Then we can use the remove ambience this method this function to recalculate the ambient RNA and remove the ambient RNA's contamination. Then make the convert the row count to log log two log two and then normalize. and make a normalization. So if we want to test, if we want to test the ambient RNA removal is good works or not, you can check the genes and actual hemoglobin is hemoglobin A1 expression and use protein expression. You can see before we remove the ambient RNA, we, we got all the and this gene would be detected in all clusters, but after removal, just a few of cells can be detected. Okay. So that means this removal is work. So after removal, we have to rescale, recluster our data and regenerate the clustering and the UMAP. So the next step is to remove the doublets. So we can use a computer doublet density to remove to estimate the probability of the doublets and calculate the score. So for evaluation, we can test it, and if the doublet score is enriched in any clusters, and we can evaluate with the feature plot or the in the UMAP or the bounding plot. So as these two plots, the result is shown and is not enriched in any cluster. That means any this cluster will not be enriched by the uh, doublet. So we only need to remove the 95% quantile of doublet score. So make some additional analysis. We can also assess the mitochondrial content, read counts, and the gene counts per cell, make a Bowling plot and scatter plot to make sure our quality is good. And the final question to estimate the variance explanation and which factor is the most contributed to the variance. We can use this command to evaluate the variance explanation and the plot. Then, so we can see there the majority is the majority vector factor is the clustering. That's what we want. So that's good. Okay. And okay, so that's for the exercise. So, so I think that's today's course, but I want to explain a little bit more detail about the loop browser, even though we only have two people, uh, three plus maps. So I will introduce how to more details of the loop browser. Mm, okay. So as we mentioned in the first, as we mentioned in the default setting is the default we got is a tSNE part and the graphic that's the clustering, and we can provide some um, customized UMAP into your group browser and cluster by the Seurat object. Right now, I will just want to show you just something like if we want to test, if we want to separate it by marker genes, what we can do. So I can use filter like this one. Okay, I can use a filter and if I want to select the cell with PTPRC expression, so PTPRC, this gene is larger than 0. Okay, you can see here. Yeah, if I want to select this. So there are 3,000 3,000 cells with PTPRC expression, and I want compare the in the knockout and the wild type one. So I will select the PTPRC expression in the wild type and make an assignment. So we will create a new category called PTPRC, and this cluster is expressed in the wild type. Say, okay, we created a new labeling like a PTPRC expression and in the wild type cell and the wild type data set. And then we can also make another filter like a PTPRC expression in the knockout. So we can assign the barcode PTPRC category, and then the cluster is knockout. Okay, so we got a PTPRC expression in wild type and the knockout. So we can use a separate view, like we separated, split on the category. You can see here in the PTPRC expression, knockout, PTPRC expression in knockout mice and the in wild type mice, and they are undefined. It's just not expression PTPRC. And then based on this, we can also run a differential gene expression. In here, we may use uh, locally distinguishing because we just need to compare these two. And the feature type is gene, then the calculation. Yeah, we can find out the genes. We can find out that the genes expression more in the knockout mice and in the compared to the wild type mice. So we can see in the knockout mice the gene expression with the PTPRC. They also have the complement expression, and they are very significant. In the wild type mice, it's more like an IL interleukin and the like a porcelain uh, like a collagen expression. So I think that's if you as your research need, you can choose you can make as many subset as you want and analyze, make some analysis and take pictures, save the pictures for for your publication. Yeah, that's what I want to explain more in the loop process. Okay. Uh, Thank you for your attention about today's course. And if you have any questions, please feel free to contact us. And I will expect to see you soon and with your new analysis cell sequencing. Okay, thank you.